demorar um pouquinho, tá?
Bom dia a todos. Good morning all. It's with great pleasure that we welcome you all to the second journey of graduate program in health and biodiversity. This year, themed regionalization, biodiversity and sustainability in the Northern Amazon. This event will be an opportunity to discuss fundamental issues about the region that is one of the most important natural heritages of our country and the world. This event is sponsored, organized by the Graduate Program in Health and Biodiversity and the Health Science Center Medicine and Nursing courses. The Northern Amazon is a region of extreme importance for biodiversity and sustainability, harboring a huge variety of species and ecosystems that are as essential for the health of the planet. However, the region faces significant challenges, such as deforestation, illegal exploitation of natural resources, and pressure from economic development, which put the integrity of ecosystems and the survival of many species at risk. The second journey of graduate program in health and biodiversity has as its main objective to bring together researchers, students, and professionals interested in the study of biodiversity, regionalization, and sustainability in the Northern Amazon. The event seeks to promote discussion and the sharing of ideas about the challenges faced in the region, in addition to presenting the results of studies and research carried out by the participants. We hope that this event will be a space for reflection and dialogue, which will allow all participants the opportunity to engage in a constructive debate on how we can work together to protect the Northern Amazon and its communities to guarantee a fairer and more sustainable future for all. We thank you everyone for attending and wish you a productive and enriching event. Ladies and gentlemen, we invite the Director of Research of the Dean of Research and Graduation, Professor Dr. Leila Braga Ribeiro. We invite the Director of the Health Science Center, Professor Júlio César Frauobi Aquino. We'd like to invite the coordinator of the graduate program in Health and Biodiversity, Professor Dr. Gabriele Mendes Lima. And as a representative of the organizing committee of the event, Professor Dr. Gabriele de Souza Rocha. To opening the event and welcome everyone, we invite Professor Gabriele de Souza Rocha.
Bom dia a todos. Gostaria muito de agradecer a presença de vocês, a participação de todos no nosso evento e falar para vocês que a gente se dedicou bastante para que esse evento acontecesse. Essa segunda jornada acadêmica é um evento, uma edição internacional. Vocês podem ter reparado já algumas mudanças que a gente fez ao longo do evento. E nós esperamos que todos vocês possam aproveitá-lo, tá? Foi feito com bastante carinho, com bastante cuidado. E eu passo a palavra agora aos outros representantes da mesa para que a gente possa ouvir um pouquinho sobre as perspectivas e as propostas do nosso evento. Tá bom? Um bom dia a todos.
We invite the coordinator of the graduate program in Health and Biodiversity, Professor Dr. Gabriele Mendes Lima, to speak.
Bom dia a todos. Sejam bem-vindos à segunda jornada acadêmica do Programa de Pós-Graduação em Saúde e Biodiversidade. Em nome do PPGSBio, quero parabenizar a comissão organizadora pelo empenho e desafio de criar um evento tão enriquecedor como este, que proporciona a qualificação acadêmica dos profissionais e estudantes da área de saúde e demais áreas ativas, que permite que os pesquisadores consigam divulgar e disseminar os trabalhos científicos realizados nas nossas instituições de ensino superior. Como coordenadora do curso de Programa de Pós-Graduação em Saúde e Biodiversidade, gostaria de salientar a importância da organização desse tipo de evento na região, para aumentar a visibilidade das universidades da nossa região norte e para valorizar os pesquisadores. Além de frisar a importância para os profissionais, quero ressaltar a importância da participação desse encontro com vocês. Entendo que, mais do que a obtenção do conhecimento, a presença de vocês aqui hoje demonstra o interesse científico, o apoio e o reconhecimento dos trabalhos realizados dentro da área de saúde e áreas ativas. Para finalizar, gostaria de desejar um excelente evento a todos e agradecer o convite de participar dessa mesa de abertura. Muito obrigada.
With the word, the Director of the Health Science Center, Professor Júlio César Prologo, aqui.
Bom dia a todos. Good morning, everyone. I would like to thank all the speakers that are listening to us and the ones that are also seeing this journey through the YouTube streaming. Thank you. Muito obrigado a todos, obrigado aos participantes, obrigado àqueles que estão nos assistindo pelo YouTube. E é um prazer enorme poder, neste ano, ter mais essa jornada. Esperamos grandemente que seja um evento que venha a crescer anualmente e que muitas pessoas venham estar interessadas em participar das ciências básicas e de pesquisas voltadas ao contexto da Amazônia. Muito obrigado.
We invite to speak the Director of Research of the Dean of Research and Graduation, Professor Dr. Leila Braga Ribeiro.
Olá pessoal, bom dia a todos. Primeiramente gostaria de agradecer o convite de estar aqui. Hoje estou representando a Pró-Reitoria de Pesquisa e Pós-Graduação, mas também sou professora do programa. Então eu fico muito feliz em a gente poder reunir os participantes do curso, professores, alunos, e também divulgar para os alunos da graduação o nosso curso e trazer temas interessantes para que a gente faça uma discussão. Que eu acho que esses eventos a gente deve aproveitar exatamente para isso, para a gente discutir ciência na nossa região e levantar temas que são interessantes para a pesquisa e que devem ser aprofundados. Muitas vezes são discutidos fora daqui e não na nossa própria casa. Então eu acho muito importante a gente fazer esse tipo de evento e parabenizo a todos, a coordenação, a comissão organizadora e desejo um bom evento a todos, que a gente possa aproveitar bastante o dia de hoje.
Thank you. At this time, we ask our guests from the head table to place in the first row of this auditorium to attend the opening conference. Thank you. All right. So now, in this morning, we'll have 3 lectures by 3 international researchers who will be presented through a brief reading of their latest resume as they present their speeches. Ladies and gentlemen, at this time we have the opening conference with the theme, The Impact of Omics Techniques on Chemical Ecology, which will be delivered by Professor Dr. Norberto Peporini Lopes from USP. Norberto Lopes is a pharmacist, master in pharmaceutical sciences, doctor in chemistry from USP, and holding member of the Brazilian Academy of Sciences. He's currently professor at USP, coordinator of the Research Center on Natural and Synthetic Products, and of the Organic Macromolecular Mass Spectrometry Center. He's a member of the Brazilian Society of Pharmaceutical Sciences, the Society of Mass Spectrometry, the Brazilian Society of Chemistry, SBQ, and the Brazilian Society of Pharmacognosy. He's also a member of the American Chemical Society, Society for Medical Plant and Natural Product Research, and fellow of the Royal Society of Chemistry. He has published more than 480 peer-reviewed journals and has received 10 awards, especially the B.R. Mass Medal, the Fernando Gallenbeck Innovation Award from SBQ, and the Jeremiah Knowles Award from the Royal Society of Chemistry. Works in the areas of chemistry of natural products and mass spectrometry with emphasis in chemical ecology. To open this event, we invite Professor Dr. Norberto Lopes to be presenting his lecture. Professor Norberto, can you hear me completely?
Yes, thank you for the kind words. It's a great pleasure for me to be with you and divide this section with some important colleagues that probably we can improve more this discussion. And I will try to start my presentation, then will disappear for me. If I have something wrong, just call me, okay?
Okay.
Thank you again to invite me. And let's see if I will do in the correct way.
All right, so the floor is yours.
Thanks a lot.
No problem.
Okay. Did you get my imaging?
Yes.
Okay.
Yeah, now it's okay. It's working.
And the idea was to try to show you a little bit how we can search with the new biological activity compounds through mass spec, but based in chemical ecology. We have different strategies to look for new molecules, but today I will try to combine a little bit. If you have an eye for the biological interactions, try to understand them and later on decide for any kind of biological activity. Let's see how was this way. First, I want to show you where I am. I work at the University of São Paulo in Pharmaceutical Science School of Ribeirão Preto. It's around 300 kilometers from São Paulo. And this faculty starts more than 70 years ago, but it's nothing for my colleagues from Europe, of course. But for the Brazilian way, it's almost 100 years. What is, for us, it's a long journey. In 1966, our group, the NPPNS, start to work. We keep the same idea until today. It's several research from natural products, mass spec, marine organisms work in the same place. And all the fundings, the money, coming through a community box where we can keep all the activities working. When I arrived in Ribeirão Preto in 1998, I started my group here. And my dream at that moment was how I can understand some biotic and abiotic effects on secondary metabolites of plants. I asked a permission to use the original figure that we published in the Química Nova journal. That's the reason it's in Portuguese and it's not translated. But that was, after almost 10 years, the first review that we published about all these combinations. But at that time, we were just starting with LCMS analysis, at least in Brazil. And today, our group expands. We work with plants, but we work with frogs. We work with some bees. We work with some fossil material. We do research on metabolism in humans, for example. We do a lot of chemical communications between monkeys. We spread all the idea to do analysis of plants for other organisms. And the intention in this case was to see how robust can be mass spec. And that's the reason almost 6 years ago, I was invited to write the first compilation of the gas phase chemistry from small molecules in electron spray machines. And how this his history started with a gas phase. I started, as I told you, in a nature product group, but in 2000, I decided to improve a little bit more this analysis, and I was in Cambridge, UK, work with James Staunton. This guy is retired now, but he opened a company based in the lodule that he get in the lab. The main idea was to modify microorganisms that produce ionophores to see if they can start to produce new molecules or new analogs to furnish to the market possible new antibiotics or things like that. And I was invited to work as organic chemistry, because I teach organic chemistry in the faculty, to work in a gas phase project in his group. And the first molecule that they gave me was monency. Then we did a lot of work in gas phase. This was in beginning of 2000. At that time, the papers about small molecules were just few. And I started my career learning about gas phase with this man, where I'm so thankful for him. Then we are more open to try to answer questions that we propose in the review that I showed you. When I arrived in Brazil, I think the biggest challenge that we get in the beginning of the career to see how we can apply mass spec to found some biological molecules, but in this case was toxin, was caramboxine. In Brazil, or in other countries, star fruit, that we call in Portuguese carambola, can be poisonous for people who has kidneys problem. And then we had a tragic episode here in the hospital in 2001, where 15 guys were waiting to do renal clearance in the hospital. And one of the guys who arrived, his wife was sick, but the guy was farmer from carambola. His wife didn't like carambola, then no problem. And he was trying to be happiness with the other guys and offer carambola for the people, and it was tragic. We had like 50 persons in very drastic answer of the contact with star fruit, and 15 of them died. And then the guys asked me to help in the isolation of this molecule. The problem is the molecule, it's very, very diluted in the fruits. For you to have an idea, it was necessary around 20 kilos of dry fruit. And remember, more than 7% of star fruit is water. It was necessary 20 kilos of dry, lyophilized fruits to get around 2 milligrams of the toxin. And all the assays at that time were done with classical bioassays, guided by biological activity. What is not very nice. That's the reason I prefer to use a picture here instead of to use a video. But they usually supply the extract in the head of the animal to see if they have some, in this part here to see if they start to have any confusion, mental confusions, and go to death. And for us, we propose at that time, based in a first observation of American groups, that could be a small molecule, and then you build a strategy based on mass spec to enrich fractions with some molecular ways that can be candidate of these toxins. And we had success. We found the molecule, this new molecule, number 1. If you look for this, you can realize that this is a kind, depend of the point of view, depend the way that you look for the molecule. Some people call this as a phenylalanine derivative or a tyrosine derivative. To be honest, it's much more the second possible, but it doesn't matter. And what we realize, we have local information that the people who has trouble but drink star fruit juice, kidneys problem, but don't eat the fruit, drink the juice, usually don't have effects. And then we decide to check, and we found in the juice the closed form of this molecule number 1. And then we synthesize the molecule number 2, and we give to the animals, and no biological activity was observed. Why? Because if you look, this carboxylic acid here and that one here, these structures look like botanic acid. That is one no molecule, very no molecule that block GABA receptors. And then we start to do this work, continuing this way. We publish this paper, I think 10 years ago or something like that. It was a long journey. And later on, because the mass spec is improved, we start to ask if the molecule can be really produced by the fruit or for endophytic microorganisms. And we had success. That's the image where the microorganism lives. You can see where you have more microorganisms, you have more toxins also. Now we know this. We didn't publish already, but we know that it's that's the reason also why it's so diluted in the fruit. And this was subject of discussions in Nature, in journals like that, or Science, sorry. But when the important point for me, sorry that's in Portuguese because the letter, it's in this paper you also claim how to treat these people. And it's amazing when you receive some letters from locals here in Brazil that call, my mother was saved because I was with your paper to the hospital, showing to the medical people how to do it. And we now also did an alternative synthetic way to produce the toxin. And now we have a patent and we are producing molecules to use in research to induce epilepsy in animal, that we can use this strategy to found for new molecules to treat the treatment. It's the opposite way. But it was my first experience how strong can be mass spec to help us to answer these questions. But then, remember, my life started with plants, and I was many times involved in how I can aggregate value to the Brazilian biodiversity. This is one of main questions in our group. And we have a problem in Brazil. Usually, if you go for any economic metrics, in Brazil, the one responsible for our biodiversity is IPEA. And the problem is you have only one equation that you use to call value to the biodiversity. And in this case, that's that question developed for Montgomery. And in this way, it's important to have a large number of research for your species. Remember, Brazil is one of the biggest countries in biodiversity in the world. When you use the number of organisms that you are identified in Brazil, compare with the applied investigations, it's almost nothing. And in this way, it's very complicated to evaluate the Brazilian biodiversity, which means that you need to do research fast. In the other ways, like you have in your state, we used to see pictures like that. They prefer to put fire in the forest instead of to check if you have other values. And then the only way now at this moment is to improve research. because we have just few data in the literature compared with the number of species. And if you have some metrics to show to the politics, they start to understand how they can preserve. Otherwise, you cannot just talk about science because they don't care about science. They care about numbers, and it's something that we keep in mind. In this way, we try from the second time in the life how to valorate the Brazilian biodiversity. And this one was a project that we do with Professor Jolie to present in the Rio Plus 20 meeting some years or decade ago. It's not so close now. But what was the idea? Usually, when you do a phytochemistry investigation, you look for an organism. What's correct? Doesn't matter. At least if you are looking for new biological entities. But another question can be, can I find something with value in nature that's already described, but can be produced by the locals? Because if you want to protect an area, I need to protect the people also. Because you cannot just put fence involving a forest to preserve. It's not like that, at least in my point of view. You need to preserve also the humans that live there. And then you need to look for alternatives. And that one we decide with this guy to do screening in a forest, in Atlantic rainforest in São Paulo state. This is a reserve area. And this reserve has a lot of pressure for the community involved, at least to collect palm trees that we call palmito, they hunting, and also many of these guys try to stole the area from the reserve to building nice place nearby the beach. And then what we do? We decide to use MAUD analysis just because it's fast to see if we can find plants. I don't care at this moment about what molecule is. It was just if we can have plants where the extracts don't have too many molecules, which means I want to see if some of these leaves, and only leaves, of course, because I'm talking about to preserve, if I decide to look for roots or wood, it's the opposite way, the impact. And then see if I have some restrict, like this extract that you have at least one major compound. We can use molecular high resolution to check in any kind of bank. And then you can do a second step to do structural elucidation. First in mass spec, if it's something really interesting, we can call this a key compound, do the isolation, check if you can do a protocol that easily produce this molecule in high amounts. What is high amounts? It's like 100 kilos, something like that. And if this theory is correct, because the first one, when we draw this way to show in the Rio Plus 20, we can call the government to support the people, transfer the protocols, and then the people who live nearby the forest can use the leaves to produce something. It was before the goals of ONU that we are discussing at these days. But the idea was the same, is to preserve an area and give opportunity for job, money, for poor local regions. And for my surprise, when we did this for the first time, the big project, we select the major plants. We cannot do this with some plants that occur 1 year, 100 meters, another tree. We need to be very dense combination. We found this species that in that area, in Atlantic forest, they have one extract. This is the crude extract, see? Has one major compound. And it's not necessary to talk too much about this compound. This is one of the precursors of vincristine. And now it's possible for these local people to produce this molecule directly. It's just take the leaves, dry, heat a little bit with methanol work, then leave it after the extraction, heating to release more the ethanol, keep quiet, very quiet, and lowering the temperature very kindly, and then you get directly the crystals. And now it's on way to go to the market. It's not a new molecule, it's not new biological activity. It's a way to introduce value for these people. And then we show this in Rio Plus 20. And also in Panama, they copy our idea, and I help the guys to start a protocol like that. And they are doing something similar there. And then you can attend one of the goals. And that's, in my point of view, the most important one in this case. Because that one keep the other ones working. Because now they take care with the forest. Because they need the forest. It's the opposite way. But another way to use mass spec is recently we helped to develop the GNPS that was really developed for Peter Dorenstein and Nuno Bandeira in San Diego, University of California, to GCMS. And then the Brazilian government, through the Minister of Health, asked us to find possible molecules, very cheap, very easy, if it's possible, non-toxic, to control Aedes aegypti. Then what we did, we can combine mass spec strategies to build molecular networks and cross biological activity of extracts, or in this case, essential oils, in large amounts. And based on softwares, you can compare what's going on and have some possible candidates, what save a lot of time in this way. Again, I'm not looking for a new molecule. I'm just looking for a useful molecule. It's not the same. And then we build these nets. The majority of the essential oil in this case were commercial essential oils that we can buy for the Minister of Health in Brazil in tons. And then applying all this combination, we saw for every time when one extract, one essential oil is active, the molecules start to grow, the circle, and the color going more to change to red or blue, depending on what we have, which means the LC50. And then you start to see in this combination that some molecules appear all the time when you have biological activities. But of course, the concentrations will change because it's one molecule or two or three in a mixture. And then based on this, we select 3 candidates. We bought the molecule because it's cheaper, and we start a pharmacotechnical protocol to introduce this in the water. And in December, last December, we gave to the Minister of Health in Brazil the first big how do you call this? I forgot. We gave him like 30,000 of new material to do in field. And we are very happy because it's working in field, which means that now it's an alternative. Now the government needs to call for some companies to produce in large amount for him, because we did in the university. And university, it's not allowed, and it's not correct in my point of view, to produce, to sell. University just develop things. Another case from Amazon now, it's about neglect disease. We have some work here in our biodiversity doing search for neglect disease. And again, we need to think when you go in this way, if I want to go to the market, I'm looking for molecules that can have low cost of synthesis, or if don't have low cost of synthesis, if we can get in large amount, as I showed you before. And we did a research in my PhD long time ago, in the last century. I don't like to say it, but this is the truth, with Professor Masayoshi and Professor Masu. And we found that grandizine and also veragencine were showing a strong activity against Trypanosoma cruzi. And then we decided to ask for FINEP in a project with a startup, Liquinoflora, to found not a way, we try for synthesis, but it's not really cheap, but to find possible plants who produce this in large amount. And the company was responsible for the clinical trials and the new formulations. And again, applying mass spec, we found this Piperacea. The first plant was a Myristicaceae. It's a tree. Now it's an herbaceous plant. That this is the crude extract. See, we can get the crystal directly to the crude extract. And this is very easy to farm. Then now we have some plantations of this one, where we can get this in large amount. And we are doing the clinical trials. What's the problem for us in Brazil? Because now we need a lot of money that we don't have. But from the research perspective, I decided to draw here in the lab a way to produce images to understand if the formulation was really good for the heart, because we need to spread in all the heart. And in animal, we realize that we have a small problem that we need to fix. This is the image, if it works, that we build in the lab. This image is supported by LCMS and computational, like in Star Wars, the way that we draw. But you vectorize every single dot here is one LCMS MS analysis. As you can see, we can go with this formulation to the heart, but not all the heart. Then we need to, you have the biological effect if the disease is here, but if it's another part of the heart, we need to change a little bit. But this is a way that we did in the past. But now I want to talk to you about to understand more the chemical ecology. That was my original proposal. And I will go fast because I have already talked 20 minutes. Then see this plant. Professor Otto, who passed away 10 years ago, sorry. He was an amazing naturopathic chemist. He did a lot of hypotheses and theories. And one of his theories is theories of borders. If you want to see the maximum of production of cytotoxic compounds for a plant, it's better to don't look in the typical environment where the plant grows, but if it's possible, behind two biomes. In this case, this is the typical biome where Arnica da Serra grow in Brazil. See, you have some cesterpine lactones here, not so high. But when you go with these plants in the border of the Atlantic rainforest and the Brazilian savanna, amplify all these molecules. And when you take the seedling from this area and introduce here, and a seedling for here, and introduce here, the secondary metabolite change, then, which means that the profile is an answer for the biotic effects. What I proposed in the first review in the beginning of this talk. Then we decide to understand all these compounds and one of these minor compounds. Then I will return to that one later. This is a flavonoid, but second time, just a fast information. This molecule, the same molecule, occur for all individuals from Arnica in all areas, in all plants of the genus, and also for the genus who live in savanna nearby, has also the same molecule. It's not the same class, it's the same molecule. But then, keep the attention here. That's the animal with a kind of one of the cancers that we work in the lab. And after the treatment with one of these minor molecules which appear, see, the same animal. The problem is, we still work with this molecule because they are very toxic for the liver, like the Taxol was in the beginning. But it's one way. We decide first to think about the ecology, how cytotoxic molecules can come in through implants, to later decide about the assay. Thinking this molecule, we apply. I don't go through the chemistry now because I don't have too much time. Just we do a lot of kind of strategies to do mass spec. We combine different platforms, but this I can answer in later. To see, Professor Semi, who was the major biologist who worked with these plants, we were in the field many times, and all the time said, Beto, Lichenophora need all these plants in the Brazilian savanna in high altitudes. They need a chemical barrier. Otherwise, they cannot survive because the environmental and because they have a specific strong area of UV light in Brazil. Remember the molecule? all, see several colors, which means every single genus that we analyze, all the plants, has the molecule. That's that one. And thinking history, we did a lot of camps. This is just some genus that we talk, doesn't matter. We decided to do multi imaging. And as you can see, all the C flavonoids are in the top of the leaf, exactly where the professor sent me proposal. I don't go in all the deep, the other ones has randomic distribution, but the end of the history is that paper was published in 2014 as the first evidence of natural chemical barrier to protect from UV light. And then based on this knowledge, we decide to check the bees who collect specifically these molecules. Go fast. We did chemistry of all these nets, things like that. We realized that they collect some specific molecules from the plant, transformed by the enzymes, and this was the origin of this new company. Every step that I showed you in the lab, when you have things like that, one of my students started a new startup. We had 7 startups in the lab. Just one was done by me, myself. That one broke. The other 6, the guys are going very well. And also we use all these things to teach local people to produce, in this case is the bee project from the cosmetic company. They teach how people in small farms, in small areas that we call assentamentos in Brazil, to produce this. And then they have an alternative. This is the net of the bee, each for the kids and for the adults, to produce. It's one social work that our group and the companies who grow from our group do to transfer money for small communities. I think I don't have more time. I prefer to stop with this message. The other one was combinations for marine data and frog data about how we get success in some other points. I want to just jump directly to save time. This is several other examples. Doesn't matter because it's just examples of the same history. But I want to just jump in to these final words. We can do science. We can do science for looking for different molecules, but science can change people's life. And in Brazil, this is really important. It was amazing how the university in Brazil was fighting for in the last 5 years. No respect with the science. Doesn't matter. The life needs to keep walking. Don't look for the past. Just look for the future. Think that we can modify the people from the life who live nearby us. We need to try. I love this image that I saw once painted in one apartment in New York. Love is the answer. We don't need to fight more. We need to try to find solutions based on science. I want to ask all my students that help my career, my wife, and my 2 best projects, Kawe and Luquinha. Thank you for your attention.
Thank you, Professor Norberto. On behalf of our university, we thank you, first of all, because you are here with us in the northern Brazil, Roraima, and also because of your beautiful, brilliant presentation. Thank you. Só lembrete, quem quiser fazer perguntas, por favor, escreva, pode ser em português, e a gente faz a tradução aqui para eles, tá bom?
Sim, mas é depois, né?
Como? Isso. After all, yeah. After the.
Agora now I need to turn off my camera and keep Philip, okay?
All right.
Okay.
So we are gonna be back in probably an hour and a half with 2 more presentations, okay? Stay with us, Professor Norberto. So to present the conference themed Nature through Molecular Diversity Arising from Co-Evaluation with Pathogens, provides AMPs as alternatives to conventional antibiotics and valuable contributors to the arms race against pathogens, we invite Professor Dr. Philip Bullitt. Dr. Philip obtained his PhD in 1984 from University of Lille, France. He had a progressive position of team manager at the laboratory of the Nobel Prize 2011 in medicine. He was chief scientific officer of a Swiss biotech company specialized in venoms. He has published over 165 scientific contributions in international peer-reviewed journals, including 21 book chapters and reviews on bioactive peptides. He was co-founder of Natural Peptide to Drugs Congress. He is also the co-founder of 2 biotech companies. He is co-author of 13 patents in the fields of innovative antibiotics. He is deputy director of a French research network on the multifunction of antimicrobial peptides since 2008, and he is director of the BioPark, a platform that's dedicated to life sciences and hosting public and private research. So Dr. Philip Bullitt, thank you for being with us. The floor is yours. We're going to have questions after the 3 presentations, okay? And a discussion time. Thank you.
Thank you for this nice introduction. So can you see my screen, my presentation?
Not yet. Could you please share it? It's not on the screen yet, okay? Yeah, now it's coming. It's coming.
It's okay?
Yeah, now it's okay. Thank you.
Okay, so thanks a lot for this nice introduction. And I would like to thank the organizer for giving me the opportunity to exchange with you. So my presentation will be rather different from the previous one because my interest is more in peptide and proteins, but the aim or the objective of my spirit is always to look in the biodiversity to see if it's possible to find alternatives to conventional chemical molecules and to find bioactive compounds. So my main focus on the bioactive molecules is regarding the development of antibiotic resistance, which is a really big problem. So my presentation will be divided in different levels. First of all, I will introduce the public health problem of antimicrobial resistance. Then I will go through an example of bioinspiration to find new natural antibiotics. And this will be illustrated through one project we are developing since many years, but we are now really inside the project to deliver a lead as an antibiotic. And I will give you also some take-home messages, including conclusion and perspective, and finally acknowledgement. So, sorry, whoops, I need to change my pointer. Okay, can you see my pointer now? Hello, can you hear me?
The pointer, yes, the pointer on the...
Okay, so, the problem of antibiotic resistance is mainly because of the fast acquisition of resistance. So the first antibiotic was discovered in 1928 by Sir Alexander Fleming. This is penicillin. All of you heard about it. It's coming from a fungus, and it's a beta-lactamine compound. And at that time, in 1941, it was considered to be the solution to treat all types of infections. But now we know this was wrong. Many different antibiotics were developed, and now we are really facing a huge need in new antibiotics because all the different families has developed, or bacteria has developed resistance against almost all the different chemicals that are available as antibiotics. And the main reason is because of the use of the antibiotic in general, Generally, has an prescription of antibiotics by the clinicians. Patients are not always following the prescription, so they stop the treatment before the end. Then you are also an overuse of antibiotic as growth factor in the production of animals. And also there is poor infection control in hospitals. And you heard about the nosocomial infections that increase and are still at high level in the hospitals. And also you discover that through the COVID that you need to clean a lot your hands and to avoid to spray in facing people. This is a way also to promote antibiotic resistance. And of course, one key issue is there is a lack of rapid laboratory tests. All those phenomena bring the problem of the antibiotic resistance. So how is the spray, how does antibiotic resistance spread? There are several ways, but the first way is the hospital, and also the behavior at home, and also the spread is through the animals and the meat you are consuming, and then the release of the waste coming from the domestic use in the water, in the sea, in the rivers, and then also the release of the waste from the animals in the soil, meaning that you have a lot of resistant bacteria going through the water or these kind of things. So now there is a really urgent need to reduce uncontrolled use of antibiotic and to find solution to get new antibiotic and new strategies. So in 1921, so it's very recent, the World Health Organization, WHO, published a report classifying the different bacteria that are urgently to be treated. And you have three major levels: critical priority, high priority, and medium priority. So this is, it's almost all the bacteria that human or animals and environment are facing. So it means there is really a urgent need of antibiotics to treat those resistant bacteria. And those bacteria that are resistant to any antibiotic are really have a huge consequences on human health. And if you see now in the literature, there is an emerging yeast candidate, Candida auris, which is really facing increased mortality in states and will spread all around. So this is also a report on coming from 2013 and a recent report, updated recently 19. And you can really see that the expected number of illnesses and deaths exclusively in states was considered to be 2 million in 2018. Now it's more than 2,600 million, 2,600 million of people suffering from illness, and the number of deaths increased twice in less than 10 years. And this is all around the world, as you can see here. And now it's considered that the antimicrobial resistant deaths could catch in the world up to 10 million of people in 2050, more than the number of people that will die from cancer. So there is an urgent need. And what kind of molecules are now in the pipeline of new drugs to treat infection, antibiotics, for example? This table is a very recent table that was published in last year. And as you can see, the number of molecules that are available is really limited. It's 252 molecules. They can be part of direct-acting small molecules, so like small chemicals. You have also antimicrobial peptides. I will more discuss on that because this is my interest. There are many reasons for that. The main reason is that those peptides are acting in a different mode of action than the classical chemical antibiotics. You can have a combination of molecules, small molecules that already bring resistance to bacteria, but they can be combined to something that will prevent the acquisition of resistance, meaning that the bacteria will be sensitive again. And there are some repurposed agents. For example, there are some compounds that were used for animals that can be used, readapted for human. The way of formulation also may also be contribute to deliver the compounds to the bacteria without generating resistance. And also there are new approaches, such combination of antiviral agents, biologics, decolonization agent, meaning preventing the formation of biofilms, immunomodulators, such as the treatment with vaccination, microbiome interaction, meaning that you can use some bacteria for to fight other bacteria, and there is also possibility to use phage therapies, but this is quite difficult because in general it's difficult to get one single phage type. On phages is a mixture of phages. To get the approval, it's extremely complicated. You can also potentiate the activity of some already available antibiotics. So coming back to what kind of institutions are involved in the development of new antibiotics. So on this part of the slide, you can see there are some foundations. There are 145 institutions already involved in this kind of researches on new compounds. There are 252 we saw already compounds on the market, on the pipeline of development. For foundation, it's limited. Private industry, it's the most important number of institutions involved in this development because of the money. But also there is a key issue in the laboratory, in the academic laboratories, because this is in general where you find the hits. Then the private company are more interested in developing the lead or to the preclinical stages or to the clinical stages. So let me give you an example about academic lab, because we are involved in this, and also on the antimicrobial peptide here, and the academic lab are more involved in these 3 stages of the development of therapeutic. So now my presentation will be directly linked to a bio-inspired project. And it's far away from what the diversity you have in Brazil. It's coming from the sea, but it's coming from the sub-Antarctic. So at the end of this South America. So you have a sub-Antarctic here, and you have a small island here, which is called the Crozet Island. And there is a specific bird living on this island, and this is a king penguin. And on those 2 pictures, you can see that the king penguin is father is giving the food to the chick. You can see here on this yellow part that part of the food coming from the stomach of the king penguin father's stomach. And it was observed by the people working on the Crozet Island that they preserve food in an aspect that is really pleasant for the chick to eat and not transformed food. So this observation was published in Nature in 2020. And as you can see here, the strategy developed by the mother and father of the king penguin was to keep some food in the stomach. But according to the duration of the hatching, the female will have no more food in the stomach. So she has to go somewhere to find food. But the male is keeping food in his stomach. And then if the female is coming back at a time quite early, she can transfer the food to the chicken. But if she is also, or if it's a little bit too late, the male can feed the chicken, and the female then can come back, can use after she came back, she can give again food to the chicken. If the female is not coming back, then the male has to give conserved food to the chicken. If this is not happening, the egg will not, the chicken feeding will not be available, and then the chick will die. So there are several obligations for the parents. The swimming to collect food now with the increasing temperature of the climate, the female has to go very far from the site of hatching, and usually it's 500 kilometers away from the laying area. And so the female is incubating only one egg, and the male and female has to feed the precious chick because there is only one per year. And the fascinating strategy is that the male retained undigested food, and the observation is the microbiota found in the stomach content was stressed or died. So we looked at that time in Strasbourg what was present in the stomach, in the food preserved in the stomach. Can we find some antibiotic controlling the microbiota? This is what we done published in 2003 by Cécile Touzeau, and for that we use a combined strategy of MALDI, of HPLC and mass spectrometry. As you heard about just in the previous talk, mass spectrometry is a key strategy to identify and characterize molecules. So what we did is we screen 60 milliliters of frozen food samples, do HPLC, and isolated some fraction, tested those fractions against a reference bacteria, and found that several fractions were positive, active against the selected bacteria only in the food coming from males preserving food, or at the medium, in some males that have a food all preserved but not totally preserved. So this is what you can see here. And then we did mass spectrometry and try to identify and characterize those compounds active on the different strains we selected. And we found two different compounds, two peptides differing by only one single amino acid, and we found that this second one here with an arginine was more efficient against the bacteria than the first one. Then what we did is we compare the sequences to what was already known in the databases, and found that those peptides are typical beta defensin that are present in birds, but also in different mammals. So what next? What we did is we ordered synthetic molecules because we were not able to produce naturally. As discussed before, it's important to produce molecules. So we did that chemically and tested on different bacteria, gram-positive bacteria, gram-negative bacteria, yeast strains and fungi in a liquid assay we developed in-house, and which appears to be is reproducible and sensitive. And we found, some interesting activities against pathogens that are of interest for birds, but also for humans, like Staphylococcus aureus, which is one of the most frequent nosocomial infections in the hospital. And E. coli, Escherichia coli here, it's a reference strain, and the infection with Escherichia coli is very important in humans and in animals. And we found also a very interesting activity against Aspergillus fumigatus, a fungi that is a problem for birds and humans. Then this is the summary of the results, and you can see here that against the Aspergillus strain, the activity is not to inhibit the spore germination, but inhibit the reproduction of the spores of Aspergillus. It means it controls the reproduction. And also an interesting feature of those compounds, they are still maintaining activities in high salt concentration, the salt concentrations that you can find in the stomach of the of the king penguin. Next step was to understand the scaffold of those molecules to be able to see what are the amino acids involved in the important for the bioactivities. This is something that we call structure-activity relationships. This was made by NMR analysis. As you can see here, these molecules are composed of beta sheet structure on a small helix. So this is typical from the beta defensins from human and from birds. So having in hands that, we were trying to raise money, because if you want to develop a hit in leads and then to go in a preclinical trial, you need to have money to optimize the compound. So we raised money very recently in 2021 through the program with the Institut du Médicament de Strasbourg. It's an institute for drug of Strasbourg. And we also raised money last year, at the end of last year, with the French agency, AINE, which is financing research in academic fields. And you have the main strategy is reported here. We are mixing molecular bioactivity screening, bioinformatics, different omics, and different techniques of biophysics to go inside the mechanism of action of the molecules. Because the key issue, if you want to develop a compound as a drug, you need to understand how it works. And if you understand how it works, if you have different molecules, then you can select which one will be the best to go right to the lead optimization. So the first, I will only discuss this first project here because this is ongoing, and the INR here is only started last year, at the end of last year. And this project is managed by Dr. Fabrice Bertil from Strasbourg. So it is divided in two work packages, identification of new defensins, and then screening of bacterial responses to the sphenicins, the name of those defensins. So here you see what we found using bioinformatics tools, the genomes, working on the genomes of the king penguin and of other birds. At the beginning, we found 8 penguin defensin, but now with the refinement of the technique of sequencing, genomic sequencing, and proteomic sequencing, de novo sequencing, we have enhanced 25 identified sequences with different amino acid composition, arrangement, but always the same scaffold with the 6 cysteines engaged in the same motif. And so this is essential for us. Interestingly, we find one large defensin combining 2 tandem copies of the same motif of cysteine. So we have now enhanced those kind of compounds and in silico. And then we have to see where those compounds are produced and if those compounds are active. And for that, we need to have a bio-guided screening of the molecular diversity of penguin defensins within the tissues. So we collected several tissues, and among those tissues, this is what we mainly are focusing on, the lung, and of course, because it's coming from the gut tissue or the stomach, we are working on the gut, different parts of the gut tissue, also the kidney and the tongue. As you can see here, we found the different sequences we observed by in silico in the different tissue following mass spectrometry analysis. So we have now those 25 penguin defensin identified according to sequence similarity with what was predicted in silico. This is something different from when you can recover from the different tissue. So now we are screening extract from tissues using bio-guided strategy. So this is what we are running now. So the different tissue, then solid phase extraction to pre-purify the compound, do protein peptide quantification, do the off-gel proteomics, bottom-up proteomics, meaning that all proteins in total are digested by enzymes, and then the digest is injected in the HPLC, coupled to a mass spectrometer. In this strategy, what we did also, we did not couple the HPLC to mass spec, but we collected the fraction by hand, and in order to have fraction isolated and to test in the bioassay. Okay, this is what is here. In the physiological condition, without salt and in the presence of salt, and we selected 3 major bacteria for their interest in human and animal pathologies. So this is what we observe. So as you can see, the profiles are totally different. So we have a lot of peptides in the different extract. Now we are running the bioassay on each isolated fraction. So we have the entire peptide, not the digest. And the results are from this morning. We have more than 15 isolated compounds with properties from the different tissues. So I cannot give you more information because we are still running the experiments. In parallel, what we did is we screen the activities on the different on selected pathogens according to the WHO. So priority 1 and priority 2 in a specific hospital condition. All those strains are isolated from patient isolated, so meaning they are well referred in specific national center for that. We know the genome of those strains that are resistant to all the different antibiotics. So as you can see here, we are always working in extremely stringent conditions, the strong conditions that are fixed for the hospital. This is crucial if you want to develop something for human treatment. So we selected Escherichia coli because this is one of our issues. We also selected Pseudomonas aeruginosa because this is an infection frequently found in cystic fibrosis patients. And we are also working on the fungus Aspergillus fumigatus. Okay. Next step is we have to understand what the peptide and protein, the defensin is doing on the target bacteria. This is again using bottom-up proteomic extraction, digestion, injection in mass spectrometry. So you can see that mass spectrometry is essential. So this is still running, so I cannot give you more information, but this is a key issue to see the modification of the physiology of the bacteria in response to the peptide. This is one point to elucidate to understand the mode of action. Now is my conclusion on next steps. So now the next step will be to go through the second project to be able to have an optimized lead and to have a document or a proof of concept that one lead we have will be right to enter in the preclinical phases. So for that, we are working on specific target. It's cystic fibrosis, and we are now running. We will run soon experiments on an ex vivo 3D human airway epithelium because this is where we want to go. And for us, it's better to use human solutions than going to mice or other models. But as you will see, for different other studies, like such as PKPD studies, you have to go to animal models. So this is what we are now developing in relation with patient association. And we are also, this is all what I can say. And now a perspective, we are also working on the formulation because as we would like to go to the lung, we have to develop spray. And we are also looking to develop a solution to treat the ocular infections because it's in both lung and ocular infection is the presence of high salt concentration. So for that, we need to develop some solution in peptide being in solution to apply to the eye. And we are also thinking about developing this approach to preserve food, because at the origin, the king penguin developed this strategy to keep food safe for the chicken. My final slide is to show you that this type of molecules, natural antibiotic produced by the stomach of the king penguin, is in fact a key partner of the what we call innate immunity. And innate immunity is present in all living organisms, from the gnatostome, meaning developed by something like 440 million years ago. So looking at the king penguin means you can look also in invertebrates, in birds, mammals, and human, whatever. You will always find natural antimicrobial peptides. So look in the biodiversity. Look in the biodiversity around you more. I think it's better to look where it is a septic environment or in a stress environment, as discussed previously. This is where you will find bioactive molecules. So my final slide is to thank first the impressive strategy developed by the king penguin, because this is our model, to thank the financial support agencies, and also my colleagues involved since many years, Yvon Lemao, Jules Hoffman, Cécile Touzeau, Céline Landon, Fabrice Bertil, and many others, students included, and to thank you for your attention. Thank you.
There you have it. Okay, can you hear me?
Yes, thank you.
All right, thank you, Professor Philip. Stay with us. We're going to have a round table later on, okay? So we are having only one more lecture. So now we are moving to Professor Tim Ludecker, if I'm right. Sorry. Series want to talk something here. So now we're going to listen to Professor Dr. Tim with his lecture entitled Turning Toxic Arsenals into the Life-Saving Drugs, Lessons from the Venoms of Small Arthropods. Tim Ludecker studied biology with specialization in biochemistry, bioinformatics, and genetics at Technical University of Braunschweig, Germany. Throughout his studies, he was a student of Professor Miguel Vences and investigated the skin toxins of European amphibians. In 2018, he transferred to the Fraunhofer Institute for Molecular Biology and Applied Ecology in Gießen. In 2021, he completed his PhD on the evaluation and biochemistry of spider venom toxins under the supervision of Professor Andreas Vilcinkas. For his PhD work, Ludecker has been awarded multiple awards and prizes. Since 2021, he is a junior research group leader at the Fraunhofer Institute for Molecular Biology and Applied Ecology in Gießen. His young lab focuses on venom biodiscovery from small arthropods to develop novel anti-infectives and biopesticides. Despite his young academic age, Dr. Ludecker has published more than 30 peer-reviewed articles on venom, has delivered talks about this topic all over the world, and is regularly appearing as a venom expert in German television. He was a representative for Germany in the Coast Action called European Venom Network, and is the current president of the German Archaeological Society. Many difficult words, right? Dr. Tim, please, the floor is yours. You're more than welcome to be here with us. Thank you. I'm not listening to you now. Your presentation is okay, it's on the screen, but your mic maybe is muted. No. We're trying to figure that out, okay? Just a second.
No, it's open.
Okay, here it's okay. I think is everything okay here with the sound, right?
Please just select the source of your mic. Between mic and camera, there is a button where you can select the microphones, the source of the microphone. You have the standard and the Realtek probably. You can change this one.
It's the second button from the mic and the camera. There is a button, okay, the blue button. So you have to click it and select your microphone, okay? Oh, almost there.
Yeah.
Yeah.
Oh?
Yes, perfect.
Okay, perfect. Yeah. Okay. For whatever reason, my headphones were kicked out from the microphone, but here we are.
Yeah.
Cool. You can also see my slide with the laser pointer, right?
Yeah, your slide's on the screen. Yeah.
Perfect. Yes. So thank you very much. I apologize for the difficulties and also for bringing so many complicated German names to the conference. I know that always confuses everyone, especially my last name. One tiny correction before we start. I'm not a professor. I'm only a junior research group leader, so I'm almost a professor. Maybe next year or the year after I will promote it, but I'm not completely there yet. It's my great privilege to be here and to speak among so many very distinguished speakers today. My work is very closely aligned to what we have seen in the first two talks, but since we are really a junior research group, of course, we are not that much advanced. We are still struggling and trying to establish our line of research very much. I nevertheless hope that it is interesting for everyone here. You already gave a very nice introduction to me, but I still wanted to just outline my CV a bit. So that's me doing what I like to do most. I was milking venomous creatures. Here it's a small monitor lizard from Southeast Asia. But before I could do this as a profession, I had to switch through several stages of my career. I started as a lab technician in the chemical industry. Then I went on to the Technical University of Braunschweig, Germany, where I did my bachelor and master in biochemistry and bioinformatics. I then switched to the Fraunhofer IME, where I'm now today as a junior research group leader for my PhD. But since 21, I am establishing my lab here. And I try to convince you now that the research that we do here is revolving around what I think is one of the most fascinating aspects of biology, of life, so to say, which is venom that we are looking at. Venom are toxic cocktails that are used by different animals to overpower prey, to defend against predators, and for intraspecific competition. And many people usually think about different venomous organisms that are very prominent, mostly snakes. But in reality, as you can see here in this phylogeny that shows all the different venomous lineages, venom is actually very widespread in the animal tree of life. You have very weird venomous organisms that you would not consider venomous at first. For instance, those of you that are more into biodiversity know that, of course, spiders are venomous, and you might even know that centipedes have venom systems. But did you know that we actually have venomous flies? Probably not, but these guys here are robber flies, and robber flies have a very sophisticated injection system in their head that they use to overpower wasps and hornets to feed on them. We have also several bugs that are venomous. We have parasitoid wasps that have venom systems. In the oceans, we have a wide diversity of venomous organisms, such as these immersion, a worm that is living in German coastal waters, and these worms have a proboscis that they can reject, and at the top of their proboscis they have these small stylets, small darts or arrows filled with venom that they use to shoot towards the prey to overpower them. And we even have venomous crustaceans, these ramipedes living in cenotes in Mexico, so underwater caves. And these crustaceans actually have also venom systems at their oral openings that they probably use to overpower prey. Not enough with all the extant venomous organisms, we even have plenty venomous organisms that are already extinct. So probably this guy here, the Northosaurus, also is or was venomous. At least when you look onto the teeth and the skull architecture, you will find evidence for a venom system that has been present there. So probably even dinosaurs were venomous. When we think about venom, we usually think about these pretty damaging effects, in this case caused by snake venoms, that are very dangerous to people. And that's why we usually think that venoms are something bad, something dangerous, because they can actually cause quite a significant amount of harm to people, ranging from neurotoxicity to tissue damage. You probably have all seen that or know people that may even have been bitten by some snakes in your family or your social circle. However, these very nasty effects are caused by venom toxins that have been refined by evolution to interfere with all the very powerful and important biochemical cascades that are running in the physiology of different animals. May it be in small rodents, which is for snakes, the prey, but even in us humans, we have the same chemical architecture that keeps us alive. And the venom toxins present in nature interfere with these chemical processes and therefore cause tremendous harm. The idea is that since these chemical processes are also sometimes involved in the development of diseases, that we can use the same molecules that cause this harm, we can, so to say, repurpose them to employ them as medicines. And this has actually a quite long-lasting research history. The most prominent case actually stems from Brazil, and it is coming from this tiny snake here, which is Bothrops jararaca, one of my favorite snakes. It's a lancehead, and these Bothrops snakes are very hemotoxic, and one component in their venom causes a massive decrease of blood pressure. And the component responsible for that is teprotid. And this teprotid peptide has actually been developed into a very small drug called captopril, which emerged as the blockbuster drug used to treat high blood pressure, congenital heart disease, and gave rise to a multitude of very successful drugs that are now prescribed probably to every third European and American person in the world. So this snake gave rise to one of the most important drug classes known, the ACE inhibitors. And this really shows that venom components, although they can cause tremendous harm, have the potential to really also save lives all along. And this is what my lab actually is trying to do. So we try to unveil venom systems across the animal kingdom to identify the components that are present in the venoms and to use them as novel drugs, but also for other applications, for instance, as novel enzymes for the industrial good production, but also as potential pesticides. And while doing that, we usually focus on our Central European species. Of course, we also work on exotic snakes, for instance, but most of the species we work with are from Germany or at least Central Europe. So we work on amphibians, different insects, and my favorites are spiders. The most fun part when you work with venomous organisms is that you can get in touch with these I find them very beautiful animals. Here I am milking a Komodo dragon, which is the largest lizard on Earth. It is also venomous, and this really is the coolest part of venom research, getting access to these animals and interact with them. However, the organisms that we work mostly with here in Germany are arthropods, specifically arachnids, so spiders and their kin. Why are we doing that? It has actually 4 major reasons. The first one is the potency. So spider venom peptides rank among the most potent toxins known. They are usually much more potent than equivalents from snakes and scorpions, so to say. They are very potent, especially on the nervous system, which makes them very interesting drug candidates to treat neurological diseases. Another major argument for arthropod and specifically spiders is their selectivity. They interfere mostly with ion channels with up to sub-channel family level specificity, which is very rarely the case in the natural kingdom. There are also peptides, and peptides usually are not tremendously stable, but arthropod venoms are heavily cysteine cross-linked, and this grants them a very high degree of stability. So many spider venom peptides actually retain their activity after oral ingestion, which means you can deliver them as a pill, so to say, which is very important if you want to develop something into a drug. And for me as a zoologist and biochemist, the most important argument for arthropod venoms is that they are largely untapped. So we actually don't know much about them. To name a few numbers here, we estimate that there are 10 million biomolecules to be found in spider venom. So far, we know only about 2,000 of those. So 99.9% of the spider venom components are so far undiscovered, which means there is so much to learn, so much to find. And this fascination really is what makes spider venoms, but also the other arthropod venoms so interesting. The question now is, when these toxins are so interesting for applied research, why don't we know more about them? What is the reason for that? And the reason actually is due to the size. You have seen the large Komodo dragon 2 slides earlier, and from such an animal you can get large amounts of venom. However, when you look onto the small Central European arachnids like this Euscorpius scorpion from the Alps, you already see that these tiny creatures only have very limited amounts of venom, right? And this is a problem because the traditional workflow to study venom starts from an organism. You would collect the venom from it, then the venom is put onto chromatographic systems and fractionated. Each fraction is then analyzed for function or with mass spectrometry and NMR for structural properties. And in the end, we then know what each toxin can do, how we can apply it, and how it's constructed, so to say. The problem is when you have only minuscule venom yields, you need thousands of those animals to even run a single experiment. And very often venom collection fails completely, which means until very recently, we simply didn't have the means to actually study our native venomous organisms in the first place. This all very recently changed with the emergence of omics technologies, because these novel technologies gave rise to the novel field known as venomics. In venomics, we combine proteomics with highly sensitive mass spectrometry platforms, and we merge it with transcriptomics and sometimes even genomics. So we start still from venom samples and do the mass spec, but in addition, we also extract RNA from venom glands and sequence the transcript. And this combination of omics technologies allows us to disentangle the venom composition of almost any venomous organism, even from tiniest starting amounts. When we have these venom compositions disentangled in silico, we can use also bioinformatic tools to predict most interesting candidates. We most of the time do this based on hammer searches, so based on hidden Markov models, but we are currently also starting to develop artificial intelligence tools to predict the most promising candidates for drug development. And this whole pipeline that we have established here as the only research group in Germany has really the power to disentangle venom compositions and to find the best interesting candidates from a given venom. The problem is when we want to also functionally study the components, we need to access them in the lab, right? Since we cannot isolate them chemically, we also need to expand our platform with biotechnology. So once we have identified a candidate, we express it either in E. coli or via self-reproduction technologies. So we use synthetic biology to make the identified components in our lab, and then we can study their function and see whether we can develop them into novel drugs or not. In the past, we worked with a wide variety of animals. So I started earlier with amphibians, but recently, as I said, we branched out into the realm of arthropods. We particularly worked with several spiders, but also with ants and pseudoscorpions. And I will give you very shortly some examples of what we did in the last year or so. The first project I want to outline a bit stems from my PhD student Sabine, who just submitted her thesis. So I'm really proud to say that I now have my first PhD student graduating from my lab. And Sabine is looking in the venom compositions of ruby ants, small, very harmless ants that are living here in Germany in our backyard, so to say. But no one ever looked into the venoms. She implied the venomics workflow I just outlined a bit. So she milked the venom, performed the tryptic digest, and did the proteome on the venom. She also dissected the venom glands, extracted the RNA, and sequenced the transcriptome, merged everything together, and the proteotranscriptome that was created thereby was then used to disentangle the venom, both on the level of expression but also of toxin diversity. And here we see the very sophisticated bioinformatic processing pipeline she used. I'm not going into detail here, but I like to show it nevertheless, because it really shows how many tools are involved in such a very simple experiment. The venom compositions of the ants that Sabine studied are very interesting because they are very enzyme rich. So the ruby ant venoms are mostly composed by venomous serine proteases that we want to study for industrial gut production now. They also contain metalloproteases and cap superfamily proteins. The most interesting toxins in these venoms, however, are the EGF-like toxins. These are very interesting because they have only been described from Australian ants before. The EGF-like toxins from Australian ants are extremely painful, but most importantly, this pain stimulus they cause is very long-lasting. In fact, these EGF toxins from Australian ants are the peptides with the longest known ability to cause this pain. So when these ants sting, the pain is detectable in mice for up to 7 days, which is a tremendously long time for a peptide. When we looked into the diversity of the EGF toxins in our German ants, we found that we have a huge diversity of those. So from the Australian ants, a total of 5 peptides has been discovered that are phylogenetically rather distinct. However, our German ants have an equally large radiation of these peptides in their venoms, and all of these peptides are completely unstudied so far. And this really shows that even in our tiny, probably also quite boring European species, we have a huge diversity of interesting biomolecules that needs to be studied. And this is what we are doing right now. We are interested in these EGF toxins from our European ants, and I actually have a master student who just finished her work in my lab, and she used biotechnology tools to express several of these EGF peptides. 3 in total were cloned into plasmids and then transferred into E. coli and expressed, and actually quite successfully we are now able to express these EGF toxins, and functional studies are about to start very soon. Unfortunately, I didn't manage to find any bioactivity data as of today. I wanted to present it here, but maybe I can come back to you and present you the data at a later time point. We also looked into the ant venoms because we are interested in a specific type of toxins that is actually very similar to antimicrobial peptides. So it's a linear type of toxin that is very often found in ant venoms, and indeed also the venoms of our backyard ants here in Germany contains a variety of those. Phylogenetically, they belong into 2 different clades, and again, they have never been tested before. So we synthesized all of them in my group and tested them against several pathogenic bacteria, including E. coli, Staphylococcus aureus, Staphylococcus epidermidis, Pseudomonas, and Listeria monocytogenes. And we could show that actually many of those peptides have a quite reasonable effect against several of these bugs, so to say, after 24 and 48 hours. More interestingly, these antimicrobial peptide-like toxins have almost no effect on cytotoxicity, which is a very, very common side effect of AMPs, but not so in our end-venom peptides, which means that although these effects on bacteria are not overwhelmingly powerful at this stage, given that the effect on cytotoxicity is so low, we think that these peptides are an interesting starting point to dig deeper into the realm of end-venom-based antimicrobial peptides in the future. Another group of linear peptides that we have been working with quite extensively in the past stems from wolf spiders of the genus Lycosa. Here are a few examples. These Lycosa spiders have a huge radiation of AMP-like linear peptides in their venom, and again, most of them have not been studied. The few that have been studied are very multifunctional. So some are antimicrobial, others are neurotoxic against insects, others are even antiviral. One of those AMP-like peptide families is the so-called A family, identified in the venom of Lycosa shansia. And since this family has never been studied before, I decided that my master student Ludwig, who just started his PhD now in my lab, had to devote his thesis on those. So we synthesized all of these peptides from this family in my group, and we performed several in silico analysis, but most importantly, we did a holistic functional profiling against cell viability, antiviral activity, anti-insecticidal activity, and antimicrobial activity on those peptides to understand their role in the venom systems and to learn whether we can use them as anti-infectives or insecticides. The data is very simple. We tested the cytotoxicity, and we could show that all the A family peptides are essentially not cytotoxic. We also tested the insecticidal activity via injection in wax moth larvae. We could also show that these A family peptides have no effect against insects. And we also tested the effect against influenza, showing that A family peptides have no effect against influenza. That makes us a bit sad because we are quite hopeful for their bioactivity. But then we went back to the sequence and checked for similarity. And what we could found is that the A family peptides have a high similarity to antimicrobial peptides present in the skin of frogs. And these antimicrobial peptides in frog skin are, of course, very active against bacteria. So we tested the peptides from Lycosa shansia against different bacteria, and indeed, many of those had a quite decent effect and reduced the growth. So do we have now here potential blockbuster drug antibiotics? Unfortunately not. All of the tested A family peptides are way too weakly active to really be interesting for any translational work, but I nevertheless want to show these data here because it's not important what we found here. It is important what we did, because until very recently, a functional study of even a single of those peptides would have taken months to be realized. The developments in genomics, especially in the synthesis pipeline, so to say, provides us now the means to test whole toxin families, whole AMP-like toxin families, in just a matter of a few months in the realm of a single, simple, small, massive project, which shows that the venom biodiscovery really is about to be accelerating very fast. And I'm really interested to pursue further studies with different families and with different students to understand their value for translational research better. Before I conclude my talk, I want to show you one last small example of what we did recently. And this example revolves around probably my favorite example here, which are toxins from pseudoscorpions. Pseudoscorpions are a group of arachnids related to scorpions, but they're different. Pseudoscorpions are venomous. They have their venom systems in their pinchers here, and they use their venom to overpower very small insects. So this is a fruit fly, and you can deduce from that these guys are really tiny. Most pseudoscorpions have body lengths of 2 or 3 millimeters, and it would be a very difficult task to study their venom. But I have a good friend, Jonas Krämer, who is about to start a postdoc in my group, and he invented the technology to actually collect the venom from these small pseudoscorpions. And he disentangled the venom compositions of several of those, and he found that a major component of the pseudoscorpion venom are these peptides known as hecacins. Hecacins are again linear small peptides that look like antimicrobial peptides, but no one ever has the ability to study those. And Jonas contacted me and asked me whether we could collaborate here, and of course I agreed and synthesized several of these hackathons in my lab and again tested their activity to understand what they are doing and how we could use them potentially for humanity. It is essentially the same workflow I just presented with the A family peptides. So we tested the cytotoxicity and found that in low concentrations, the hackathons are very weakly cytotoxic, so to say, and even in concentrations exceeding 50 micromolar, they gain cytotoxicity. We also tested the effect in P aphids, and interestingly, we found that these hackathons have a very potent effect against aphids that are comparable to spinosad, a commercially available insecticide, which tells us that these peptides may be even interesting for the development of novel insecticides. But we also tested the hackathons against different bacterial strains. And we could show that especially one isoform of hackathon 1 has a decent activity against several of the already mentioned bacteria, such as E. coli. But most importantly, it has a very potent growth reductive effect of this strain of Staphylococcus aureus, which is coincidentally a methicillin-resistant one. So a multi-drug resistant strain, and that strain is heavily affected by our hackathon here to say. Which means, paired with the low cytotoxicity of hackathon 1 that I just showed earlier, that we have here in our component that may actually be interesting to be pursued upon in future research and maybe for development into novel antibiotics, or at least into novel tools to fight the spread of these peptides. And therefore, we are now investigating different isoforms of these. We are also creating artificial forms of hackathon 1 with engineered residues, but we are also going now into material science. We are playing around with hackathon-loaded nanoparticles and vesicles to formulate those peptides to really create novel tools to fight the spread of multi-drug resistant pathogens. We are almost done, but before I conclude my talk, I want to provide you with 3 take-home messages. The first one is that we have many unanswered questions in this field of research. We now have genomics, we now have biotechnology tools, and this allows us to study all venoms. However, given the huge diversity of species that are venomous, we are really just starting to scratch the tip of the iceberg, and there are so many species that need to be studied in the future. I also, that's take-home message number 2, I believe that venoms, especially from arthropods, have the ability to make our world a better place. I think that arthropod venoms can really create the medicine and also the agronomy of tomorrow, and really can change the way we are living and make our world a better place. And the last thing I want to mention is not really a take-home message, it's more kind of a question. When we look at this huge diversity, both on the level of organisms, but also on the level of molecules found in venoms, I really want to raise the question, how could we ever dare to ever want to study something else? I really don't know. For me, there's nothing else. With that, thank you very much, and I'm happy to take questions in the roundtable.
Thank you, Dr. Tim, for your presentation. And to mediate the discussion based on what we have heard so far from the renowned researchers, we invite Professor Luis Fernando Garcia Hernandez, who is a Colombian biologist who settled in 33, where he is a member of the University Development Pole, the multidisciplinary group in ecology for agriculture, and teaches at the Esta Regional University Center. His area of study is entomology. Within it, he is interested in arachnology. For this reason, in 2014, he traveled to the European Congress of Arachnology in Italy, where he met the Australian Dr. Sean Blemeris. Blemeris is an arthropod silk specialist. Well, for those who are here in this auditorium and want to ask any question, please direct them to the organizing team, and we are going to address them to Professor Luis Fernando. Professor Luis, can you hear me?
Yes, I can hear you. Can you hear me?
Yes, perfect.
Okay,
so
thank you.
I already sent you some questions on the chat here on this platform, okay?
Yeah, I can see the questions now. Thank you.
Perfect. So the floor is
Well,
yours.
so firstly, thanks for your invitation. And well, thanks to all for your great talks. So I'm not sure if we can maybe at the beginning try to solve the questions that were made by some guests, and afterward we can make a small open discussion to close the table. Do you agree? Yeah. Okay. Perfect. Well, we have several questions here, but maybe we go one by one, and well, some of them are directed to I think to all the speakers, and some of them are maybe only one of you could reply to them. Well, firstly, we have a question here from Maria Moraes, and she wants to know how can we create public policies for health to encourage fighting against the health issues present. I think.
Could you please repeat the question, Professor Luis? I think.
Can you hear me or
Your sound is complicated to listen.
Yeah, it's cutting. I think it's your internet. Yeah.
Okay, now can you hear me now?
Yes.
Better or not? Okay, we have a question here from Maria Moraes, and she wants to know how to create public policies to fight the health issues which were shown in your presentations. I don't know who wants to reply first, maybe Philippe or Norberto, who were more focused on the health issues topic.
The question was not clear for me because the sound is very bad.
Can you hear me well, or maybe I will use a headphone? Let me check if I use this and it improves a bit.
I don't know for the other speakers, but for me it was not clear at all.
Yeah, it was not possible. Probably you have two microphones together with you, like your computer and the mobile.
Can you hear me now?
Try again, because we had two sounds coming from you.
From me? No.
Yeah, do you have like a mobile?
No. I just have one microphone. I just have my computer. It's strange. But can you hear me now?
I think it's much better now.
Yeah. Okay, maybe it was the computer. So the question is if how could you create public policies to encourage and to improve the fight against the health issues that were presented here? Maybe what kind of promotion you make, or
so maybe I can answer regarding the antimicrobial resistance. I think it's difficult to enter in the policy in general because, in fact, if you want to develop a new antibiotics, it costs a lot. The policy is to reduce the use of antibiotics. So for a private company, if the production has to be reduced, then if the cost to develop one is extremely high, then if the conflict is it reasonable for a company to produce an antibiotic that will not be used, or the policy is requesting to decrease the use. So when I was on a roundtable 2 weeks ago, one of the discussion was we have to fight against the antibiotic resistance. We have to find a solution anyway, because we cannot avoid the antimicrobial infection with bacteria, fungi, and yeast. So you have to find a solution. And you have to push the policy to, okay, the cost is high for a compound, but is less as the cost of the consequences on human being. And if you can orient this policy to the private companies that mentioning that, okay, you have other alternatives, because the company is not only working on antibiotics, they have a series of compounds on other pathologies on this kind of thing. But the resistance of bacteria is increasing, it's going faster. So they should be convinced that the cost is not the cost of a compound, but it's the cost on the society, because it's human being. It's also the daily cost, because when you are arrested because you are illness, it costs a lot to the society, and all those consequences has to be taken into account. And this is not what was done before. But the COVID situation is something like a very useful clinical trial, even if it's something wrong, but it's worldwide. It's a test worldwide that we were totally blocked by a virus everywhere in the world. So this situation, very bad situation of COVID, must be something interesting for the policymakers to think about. Okay, we have one worldwide trial. We do not have to do twice that. So the cost is something which is not really, the cost of the molecule is not something important, but we have to convince the pharmaceutical companies for that. And this is a huge work. This is my feeling.
I agree with Philip, and also in Brazil is even worse, because you need policies at least to have access to the clinical trials. This is another thing that we need to fight too much in our country. And I really believe that you need to make, like during the COVID time, to make consortiums of people to help, because this will be a problem, as Philip said very well, from the humanity very soon. Because the reasons are growing fast, and you don't have any real support, for at least in Brazil, for the government to take care about this. They prefer to wait the big problem in front of his face instead of to start to prevent. This in Brazil, more complicated.
I think if I can add something also, it's the same for the largest countries like China, India. It's the same problem. Exactly the same. And with the traveling of people, the world is extremely small. And this is a huge need.
Okay, I don't know if Philip wants to say something. Sorry, Tim wants to add something, or do we move to the next question maybe?
I would just add to that, when I speak to people from companies that come over, they always mention the huge price tag as a major barrier for their side. I think what policymakers could and should do is to think about how we, from governmental side, can at least reduce this price tag a bit. They will not give money into it because governmental money is it's given in basic research programs. But I think there should be ways to support companies that could actually help in this important task that is developing new drugs, so to say, by reducing taxes, whatever. Like we need economical tools to support and to motivate the companies to dip their toes into this research and to get involved, because from their own money they will not do it, only at a large stage, and there need to be political arguments to support the countries everywhere to actually do this. And I feel this is not at least in Germany, our politicians are not considering that. They think that the companies are extremely wealthy and they can pay everything from their end without any support, but that's clearly not the case. They will not do that.
Yeah, great, perfect. So I don't know, do you want to add something else, or do we move to the next, maybe, to the next question? Okay, yeah, great. There is a basic one here, which, well, if the ones from you who wants to reply, It says, it is a basic question, what are the physical and chemical principles in mass spectrometry? I think this is made because we have a medical audience. So, so if some of you want to take the challenge and
in a single shot, my way.
In a single shot, maybe. I don't know, maybe Norberto, who maybe you mentioned it.
If the other ones want to talk, for me it's okay.
I don't know.
Yeah. It's not so simple, it's not so basic. Depend the weight or the chemical properties of your molecule. For example, if they are thermostable and can be volatile, you can use electron ionization hyphenated GC machines, which means that you have your molecule go into the gas phase, you shot an electron, take it off, and then you have a cation radical ion, which means normally you have molecular ions. That's a molecule with a charge with the same mass as the analyte. In electron spray, it's not so simple like that, but I'm trying to do in a simple way. You have a molecule in a liquid coming through, for example, LC. You go to the source, you spread this. It's a night kind, it's not chemical, you don't have temperature now. And then when you have the drops, you apply a charge that usually is enough to do the dissociation between H and OH, the water dissociation. And then when the drops start to reduce the size by an opposite gas, you have 1 minute or 1 moment when the repulsion between the charges is more than the superficial tension of the drop. And then you release the ions from the gas phase. In this way, you have acid-base reactions or you have coordination reactions, which means that the ions that you see is protonate molecule, for example, in positive mode, which means that +1, or sodiate molecules, which means that the number that you have in their spectrum is +23. And this is just to see the ion. You have no fragments, no structural elucidation. Then you need to isolate the ion, go to a second chamber, excite with a collision gas, neutral if possible, transfer kinetic energy, increase the internal energy of the ion, and then fragment. And then you have the second MS, second analysis, and then you have the first fragment profile for you to work. That's the major. You have MALDI when it's solid. Then you put your analyte inside of a liquid that's not really liquid, it's not really solid. It's like the glass. And then you have this more thickest. And then you, inside a laser, shot laser, explode, and then go to the gas phase. In that one, the majority of the reactions, it's not the unique, but the majority is acid-base reactions. But you can have also excitation that take one electron or think like that. But this is very fast. One idea what's the mass spec.
Yeah, many thanks for this quick lecture. Well, we have another open question. Maybe all of you can. This is focused on epilepsy, but maybe if you want, you can give a particular example. I guess something you think. And it says how something that can cause epilepsy help to develop compounds that can be used against epilepsy. So the idea is that well, I don't know if you get the question. Maybe you could reply each one of you, or maybe use a particular example that you know this is an epilepsy, but maybe we could move to other fields. I don't know who wants to reply. Maybe Philippe at the beginning and after.
No, for me it's far away from my field of expertise.
Or
so
Norberto.
I think it's better for maybe Tim can go on Norberto. Norberto can go in this. For me, it's far away.
Okay.
I can try to elaborate on that.
Yeah.
So, what one always needs to consider, a molecule that causes a disease or disease state. Of course, it is a damaging component, but this means that we can take this molecule, take it in the lab, and study it on cells, for instance, to understand how a disease works. If we have a molecule that causes a disease attack, so to say, I can put it on my neurons and can use it as a tool to study what happens in the neuron during epilepsy. And this knowledge I can then use to fight the disease. This is how a disease trigger can be used to fight the disease. But a molecule that interacts with a receptor that causes a disease, and I know it does it, I can study also the molecule, I can identify the sites that are reliable to cause this disease, and then I can play around. If I have a peptide, I can exchange amino acid, I can modify the properties until the stage that I create a molecule that works in the different direction, so to say. And then I can create an artificial molecule that may even have the potential to block the disease, very, very simply speaking. So it's not that easy. It takes 20 years to do that, but this is the idea behind it. So a molecule that is damaging, when I understand it properly, I can use it to understand the disease and maybe to modify it into a component to battle it.
Thanks, Tim. I don't know, Norberto, if you want to add something or...
No, in our case it was almost this idea. We found one toxin. Then we learned where the toxin blocked it. Then you use this toxin, as Tim's already explained, that you know where it works. And then you can now use the molecule as a way to induce the disease and try to find molecules that can put the toxin out and can help in this way. The toxin is not applied to treat. The toxin is one tool to help to induce in the animals for you to do biological assay with other molecules. Is that? But
yes.
He has already explained very well.
Yeah, this is the same for antibiotics. If you have a bacteria that is resistant, you need to know the mechanism of resistance and the mechanism of actions of the compound you want to develop, and you have to play with those two steps, the recognition, and this is the key issue, as you mentioned, both of you.
Perfect, many thanks. So we can move to the next question. This is directed to Philip. And it says that considering the complexity of new antibiotics and their increasingly strong action potential, can this rise to greater doubts? I think it's like greater problems as super resistant bacteria, which will be unable to be treated in the future?
There are already one bacteria that you cannot cure. This is E. coli, Escherichia coli, which is the last antibiotic, which is extremely toxic for the kidney, is not working anymore on some isolates. So we have now at least no solution for this pathology linked to Escherichia coli, because the colistin is not working anymore. And this one is highly toxic for human beings. So this is the reason why it was, until 2 or 3 years ago, the only antibiotic remaining active on this specific strain of E. coli. But, I mean, if you want to cure someone, the danger is you will kill the kidney because of its high toxicity. So we really need to find something, and with this new type of antibiotics like peptides, their mechanism of action is really very different from the ones before, because it is not acting directly on the membrane of the bacteria, but it is crossing the membrane and interact inside the cells, inside the bacteria, meaning for the bacteria to develop rapidly resistance is probably less easy than with the classical chemicals.
Okay.
This is
we have another one for antibiotics here, and it says that if the antibiotics found in the penguin's stomach could be used to preserve food, like for example, in the meat industry, could have some meat industry applications.
I mean, that was the first purpose of this molecule in the stomach. So we try effectively to promote this, but it's difficult to combine both to get money. We cannot get money working on the antibiotic, and we cannot get money to develop something for food preservation. But we already tested food preservation using seafood because of the salt environment, and we have some indication that this is something interesting, but we need to improve that and we need to develop that. But this is one issue we would like to develop in a second way. First, we need to have a therapy, to have an antibiotic for human and animal health. But food preservation is, of course, in our mind, because it's the origin of this molecule.
Okay. There is another one that is directed to Philippe, but maybe you could respond. I'm trying to figure out the question because it's a bit dispersed, but it asks about the role, for example, of the interaction between animals and environment when developing these antimicrobial compounds. But maybe you could also talk about venoms and maybe
I think so. Yeah. You
can,
yeah. I think I can talk about venom. It's linked to Brazil, more linked to Brazil.
And finally, I don't know if maybe I think we are getting out of time. Looking at Claria's screen.
Yeah, time is running for everyone everywhere.
But I think it would be interesting maybe for closing this lecture to if all of you could maybe give a brief message about the importance, maybe for medical people, of knowing the biodiversity in megadiverse regions such as Brazil and Roraima, and the development of these compounds that could improve human health. Don't know if you want looks like a closer message, maybe from.
So if I can start, I will say to you what I am saying to all my students. Just look around you. Nature developed during its evolution absolutely key features to preserve its living style. If you look around you, you will find solution for the global health, if I can say that. Just look around you. Never forget to look around you. Specifically if you are in a septic environment or if you move plants as you saw in the border of forests that are not used in that, or flowers, they will develop some stress. Then they will react to that to adapt or to survive. And if you are at the right moment, if you observe that, then you will find interesting molecules. But preservation of biodiversity is the key for bio-inspiration, and that will be the solution. I mean, human is just copying what nature did. And adapted because he has tools, technical tools for that. But the nature, you don't have any mass spec in a plant, but she developed its strategy to be still alive. So that's my message. Look around you.
Maybe Philippe or Norberto?
Doesn't matter. Can be huge. Okay. My dream is to stole the Philippe words. It's exactly what I say to students, because as the plants that I showed you and the frogs and things like that, is just look for the ecological interactions, how the animals and the plants interact with the environmental. If you think in this perspective, you will find the answers in the nature. We can do, you have the way that the industry do, like prepare extracts, and this is one way, it's not wrong, it's their way. I think in science, you need to open more the questions and look in what's going around. That's the perfect word. Philippe used very well. And it's what I really believe. Take care with the ecological interactions that you find the answer for a new essay or things like that. But look first what's going on, then decide to the essay. Thank you for inviting us.
Thanks. And last word, Philippe. Sorry. Team.
Yeah, I think my conclusion goes very much in the same direction. I mean, we humans, we are biological beings, right? And the problems we are trying to tackle that are threatening our health are also biological problems. And we are a very young species that hasn't found many answers to these problems yet, but there are so many other lineages that are so much older that went through millions of years of evolution, found solutions for those problems. So many of the things we are talking here today have been solved in nature already multiple times independently. And it's our job to look onto these organisms to see how nature solves specific problems and try to learn from that, how we can mimic that for humanity. I think this is essentially what we all are doing here, that we have presented here today. And you guys that are sitting in one of the most biodiverse regions, you have this unbelievable huge treasure trove of biomolecules right in your backyards, right? You have so many species, and each of these species has so many biomolecules that could have value for all kinds of things, not necessarily only health. But it's important that someone is going there, is looking into these chemical diversity spaces and finds these molecules and studies them. And it is even more important because we are living now in the Anthropocene. We are losing species. And since all these molecules are very uniquely found in sometimes even one species, this means that when we lose this species, we are also losing the bioresource with it. And this is the most important thing because we are losing species now everywhere at an unprecedented speed. And when we are not very fast here, when we miss this chance to study the chemical diversity across the tree of life, so to say, we may lose all these important biomolecules to treat diseases and to do all kinds of things. And I think this is the most important thing. Like biodiversity loss means loss of bioresources, and we need to be faster than the loss of these things. Yes.
Well, okay, many thanks for all of you. So I think we are on time now, right? So everything went fine.
Before we finish this session, Professor Claria wants to say something.
Okay, first I'd like to thank you all for the nice presentations, yeah. And what I say to here, we have a laboratory in our backyard, and we need to go so we don't need to go so far to investigate our biodiversity, but sometimes we have problems, yeah. That is to put people to work together, and it was just what Team Dudeka mentioned, yeah. And I would like to ask you, yeah, because you have a lot of experience, and we have a new post-graduation program, yeah, that is in health and biodiversity, but we have this problem. And it's interdisciplinary, yeah, and this is important, yeah, because we have people from different fields, yeah, and I would like that you give us some advice, yeah, to find the best solutions, yeah. I think that's it.
So I don't know if I can start, but I will start anyway. I think what I heard from my experience in 40 years in different labs, in different companies, the best chief, in this case, it will be a topic of research, is to bring around you the best competencies. No one has all competencies. So collaboration between teams with different competencies in terms of technologies or from the biological matrices up to the analysis. This is the key issue. You can never work alone. You can never work alone on the same area, but you can never work alone. You need to have a worldwide collaboration with expertise and competencies in different fields. If you want to develop a drug, you need to find the drug. It means you need to find the right biodiversity model. You need to find people aware of screening. You need to work with hospital. You need to have a veterinarian around use and a platform of technologies. All that you have to bring them together. It's a huge work for all of us. We need years for that. But this is a key issue. You cannot do alone without, forget it. It's too complicated, especially in difficult area where you are living. It's not easy to bring chemicals. It's not easy to bring things like that. You have to organize, to combine your force for that. Without that, you will not be able to compete with other people. Competition is the best thing. Alone is nothing. But competition is something will bring you ahead. Without competition, I mean, time is running smoothly. But in these terms, if you want to cure people, if you want to cure animals, if you want to save ecology, environment, you need to go fast because time is running too fast. So you can only do that if you are combining all your forces. This is my opinion.
I also would like to thank all, Dr. Norberto Peperoni, Dr. Luisa Garcia, Dr. Philippe Boulet, Dr. Tin Ludek. Thank you for all. This was a nice journey. I think for the future, we're going to just improve and increase the collaboration as Dr. Claria was trying to establish here. And I think this moment for our research is just the beginning, just the first step. As she said, our backyard has many sources of compounds, of probably different plants that we don't even know that are possible. Like ibuprofen was, it's a drug used worldwide, and the source is the Amazon, and probably we do have many others as ibuprofen, and I'd like to thank you all. We are finished this journey, and really happy because we all agree that was a nice day with interdisciplinary conference with people from different fields, bringing all your experience, and we are really thankful for all you doctors for this moment. Thank you so much.
Thank you to all. Thank you for inviting us.
I'm gonna say bye. We're gonna keep in touch for the future, okay? We're with the doors open for you to visit us also here. Thank you so much.
Many
We're
thanks.
ending for today. Thank you. Bye
Bye bye.
Thank you.
Bye.
Bye.